we can start in our again with our journey in evolutionary ecology. And what I want to start today and I guess continue tomorrow is trying to sort of merge together the two sides of what we did last week. So both the evolution part and the ecology one. And the system in which I want to study this is the one that we introduced last week, right? So this is by no means a very special system. It's not something that it's of particular importance, but it's a good study system in which to understand fundamental process in ecology and evolution. And just to remind ourselves, since it was in November, we are in December now, so we have viral particles in our system that what they want to do is to inject their DNA inside a bacterial cell, and when they find one, they inject this DNA, and then with this DNA gets transcribed and translated and produce what? Produce all the stuff that are needed to produce no viruses, and then these viruses accumulate until they sort of are enough. They start to produce some compounds that make the cell explode, and then there is like a burst of these viral particles that goes into the system. Okay, so this is the sort of splatter view of infections. Now, the kind of question I want to study is, suppose that you look at this system and somehow you want to study evolution, you want to see how these change over time. If we think about what we did last week, we were talking a lot about fitness. I mean, we were talking a bit about fitness, and the fact that you expect that the fitness of some organisms is going to increase. Right? So now, if you look at this system here, we wrote last week some equations describing the population dynamics. And so the question is, what is fitness in this case? Okay. So what are the properties that determines that can evolve and change, and what direction they have? Okay. And so the first thing we're going to do is to first imagine that, I mean, if you have evolution, what you can imagine is that you have a mutant that arises, and this mutant has some different parameters, I mean, at different rates, okay? And then you want to know the outcome of what happens if you have two different mutants, two different groups of individuals with different rates, okay? And sort of so suppose that we want to describe it. So what we want to write are some equations describing how these populations of multiple phage strains with different parameters change over time. Okay? So I'm doing this because we have to write equations now. Okay. So what equations am I going to write? Yeah, first of all, we have bacteria, right? So let's first write what are the quantities I want to describe. So first of all, we have bacteria. Okay. So now I'm imagining that bacteria don't evolve. Okay? This is not true, but let's say for now, let's forget about it. Then what other equations do I want to write? Okay. And then? Yes. That's it? Read the title. Yeah, so I have multiple types of phages, right? So here it's one type. Now I want to add another type. So what other equation should I write? Huh? The other type of phage, let's call it V prime. The other type of infection. And the other type of infection, right? Because if I'm a cell, I'm a bacteria cell, I can be infected by one phage for the other type of phage. Okay? So I have the question is definitely too much, but let's try to write it and look at all the terms we described last time. So this equation, what is the first thing we want to write? Yes, the logistic equation for the bacteria, right? So we say how these bacteria change if there are no phages. Okay? And then there is what? Yes, there are infections, right? So what we wrote last time was a term like this. You are infected by these viruses, and then if you are infected, you get term here, and then when you infect something, there are fewer of these phages in the system. Okay?
The same for the other type of.
Exactly, and then we have the same for the other type of phage. Okay? Now, in principle, I could say now. Well, let me write it here. I have P and B prime, and here I have minus P and B prime. Okay?
But right there, we're saying that they, like both viruses, infect at the same rate.
That's a good point. I mean, these are two different viruses. Why they should be infecting at the same rate? Right? They could be infected at a different rate. Right? And then what other parameters I have? We have lysis parameters, so we say with some rate eta, these infected bacteria explode. Okay? And so I have beta, eta, what's this character? And then I have the same term here, right? So I have minus eta prime, I prime, plus beta prime, eta prime, I prime, right? Then
The death rate of the virus.
yes, then we say we have the death rate of the viruses here. Oof. Okay. Good. Now what I want to do, so this is like in general a system where we have one bacteria and two types of phages. Right? So what I want to do is to do a slight change to the setting respect to last week. Because it simplifies a little bit things. And what I want to do is to imagine that, well, now this is like pretty general and pretty complicated, so we have to simplify things. But before simplifying things, one first simplification is that we can assume that we will assume that some parameters will be the same for the two types of phages. Okay? So for instance, I'm assuming that this omega is the same. If you don't like it, with what we learn now, what we will learn now, you can play and study what happens if you have different objects. Okay? Then the other things that I'm going to assume is that this phi is going to be the same. So the rate of infection is going to be the same. Okay? And what we will study is what is the effect of eta and of beta. Okay? So what is beta is the burst size, how many viruses you produce, and eta is how much time does it take from infection to lysis. Okay? Now, the first thing we're going to start is actually assuming that the etas are the same. So we have just beta. Okay? As I said, I want to modify a little bit the equation. And in the way we interpret this mortality. Okay? So in this case, this mortality was something where we were imagining that there was like a tank, and there are bacteria inside, and somehow there is a rate at which phage particles, virions are called sometimes, they become not effective anymore. Now what I want to think is a system where basically we add some nutrients, and we remove some stuff. So bacteria, phages, infected cells, etc. Okay? So I want to think of a system like this. Okay? Where now this term mortality refers to the rate at which we remove stuff. Is this clear? So if we have this omega refers to the rate at which we dilute from the system. Okay? And so as we dilute, we remove viruses, we are also removing infected cells and bacteria. Okay? So we have minus omega I, minus omega n, minus omega I prime, minus omega. Okay? So basically the idea is that we have this system where we have a tank and we put nutrients inside, we remove stuff because if we keep putting things inside, these things is going to grow forever. And when we remove also some of the cells. This is called the chemostat. Okay. Good. Now, yeah? Oh, I removed twice. This goodbye. Okay, so now we have this system here, where the only difference between the two types of viruses is what? Is the birth size. Okay? So how many viruses they produce when they infect a cell and explode. Okay. So let's think about the following. So suppose that we start our experiment. It is going to be a simulation. And we have like these bacteria, happily living together, and then there are these two viruses that arrive, right? And they have two different betas. What do you expect is going to happen? So you have two viruses, they infect at the same rate, they lyse at the same rate, they die at the same rate, but one produces 50 cells, the other one produces 51 cells. Too much excitement. Yeah.
Maybe one is going to dominate because it produces more, it's going to infect more. The cycle goes like a little bit.
Yes. Yes. So what happens is that if we it will appear behind the equations, but we can leave it like this. So this is the concentration of viruses. And here I'm imagining that there are two types of viruses. The orange virus that have beta equal to 5.2 in this case, and the blue virus that has beta equal to 5. This birth size is bigger. We start with a small concentration. What happens initially? Initially, it happens that both the viruses grow because they're infecting, but then there is a point where the number of bacterial cells starts to run out, and at that point, what you see is that their fate is completely different. Right? The one with the highest birth size are going to dominate, right? And they go to some stationary value, which is the one we calculated last week, while the other ones will decline. Okay? Will decline because they are outcompeted by the rest. Okay? So the idea is that in this case, what we see in this case is that somehow these phage have a higher fitness, if you want, than these other phage. Okay? Now, this is not particularly surprising. We are saying we have two viruses that are sort of analogous to each other. They are equal in all the aspects, except that they have different birth sizes, right? So the ones that produce more viruses are gonna have more viruses that accumulate. They're gonna dominate the system, and then the other will have always less and less and less and less, fewer and fewer and fewer and fewer viral particles in the system. Okay? Good. Okay, so how do we understand this? Now, this is pretty intuitive, but I want to sort of get an intuition about this. Okay? And so the way we get an intuition about this is that instead of focusing on what happens when we have two different viruses that, well, we can do another, let's put it this way. We can do another experiment. Instead of starting with two different viruses that are at small value and they invade the system, what we can imagine is that originally there was one virus, okay? And then another one arrives. Is this clear? Good. So we have one virus that is happily staying there, happily infecting all the other bacteria, and then there is another one that arrives. And then we want to ask, what happens to this virus that arrives? Okay? So what do you expect? Huh? Yes. So suppose that you have one virus, and that has a given set of parameter traits, etc. And then arrives another viruses. What are the possible outcomes of this? Yes, so there are two options, right? Either this virus will die or they will dominate. And what do you think it determines this? The birth size, okay? So let's look at the simulation just to, it's okay if I show the simulations behind the equations, otherwise I have to rewrite them later. Okay. All right, well, let's actually, I rewrite them later. So here what do we have? We have an original population that looks at this line, where there is host population N, there are some bacteria, they are in a fixed point, like the one we calculated the last time. So if we don't do anything, they're gonna stay there, and this is perfectly balanced, right? There are viruses, they infect some cells, and then they kill some cells, etc. This is a fixed point. And now it arrives an invader. So the resident population, the orange one, has a beta birth size equal to 5, and this one is a beta prime equal to 4.9. Okay? What does it happen? That if we start with a small population, this is going down, right? Because it's not able to invade the system. While suppose that we do the opposite, right? We have a resident population that has 5, but now the invader, these new few viruses, have an higher value of beta. They produce more viral particles per infection. Okay? What do you think is going to happen? We are going to go up. This is exactly what happens, right? We start with the population now. This one is still 5, but this one is 5.1. So they go up. Okay? And then if I wait a longer time, what do you think is going to happen? Yes, so this one will keep increasing, and the other one will be decreasing. So this is the same plot. This was up to 2,000 time units. Now if we go to 12,000, what you see is that this is growing, and it's taking over the population, and this one is declining. Right? And so this displays the other one. Okay? So a nice way to look at this, no. Let's look at this thing, okay? So let's try to look, to understand why this is happening from a mathematical standpoint, right? Intuitively, we understand it sort of clearly, okay? But we want to have a better understanding of what is going on, okay?
Yes. For example, both, like, there are two different viruses, but they have the same data. So that would mean that they would like.
That's a very good point. We will see what happens. As an intuition, what happens is that you cannot distinguish between these two. Right? That is called like a neutral mutation, in this case, which means that even if these two viruses are different, there is nothing that makes them different. So it's like if you were coloring them with different colors and tracking how they were expanding and changing, but this will be completely arbitrary. Right? There is no difference between them. Okay. So let's try to look at it systematically. Okay. So what we have is that we have our populations of bacteria. And so this is the resident population, right? We have this population that is at the fixed point, is happily staying there, So this is beta A I minus omega B minus B. Okay? So it's happening staying there to what value? Well, we can calculate what is the fixed point, right? Which is what we did last time. And in particular, what we know is that the fixed point is going to be, well, we can set this equal to 0, right? And we get that I star is equal to phi n star p star over eta plus omega, okay? And then if we substitute in this equation, we can solve for n star, and we get that n star is going to be, now it's a bit different from, it's omega, but let's write the equation. So it's beta I star, which is phi n star b star over eta plus omega, right? Minus omega b star minus phi n star b star equal to 0. Okay? So then we have this thing we remove. And so if we solve for it, we get n star, which is equal to omega over phi times eta plus omega over beta minus 1 eta minus omega. Okay? Which is slightly more complicated than last time, but is what we need. Yes. Okay? So we get that the population of the resident value of the cells is equal to this one. Okay? And then the population of the bacteria, well, the population of the bacteria is equal to this one, and the population of the infected cell is equal to this one, and then we can get the solution for V star. Okay? Good. So this is the state where the system is, where the new virus arrives. Right? And now what we have is that we have this new virus, and let's do it in another color. No, we can make it a real. So we have N, B prime, and then we have the other equations, right? And these equations are going to be beta prime, eta I prime, minus omega B prime, minus P and B prime. And here we have P and B prime minus eta plus omega. So now if we want to solve this equation, how do we do? Well, we have to integrate these equations, and this is pretty complicated, right? Because we have this equation here, but the equation of the cells are going to affect the population. So N is going to change, and then n is going to change these dynamics here. And so, again, why am I writing on the board? Okay. The point is that there is one trick that we can use. Okay, and the trick we can use is that initially the invader is at a very, very small population density. Okay. So when the invader arrives, the invader is at a small population density, which means that this term here, what we can do with this term here? We can neglect it, right? Because initially the invader cell doesn't produce a change, right, in the population of the bacteria, because it's so small that it doesn't affect the system. So this term, we're going to approximate it to 0. Okay? But then, if we look at what we are, but then, okay, this is still complicated, right? Because we have n, and we have n here. But what we can do with that n? What is the value of n where but n is not 0. I mean, this term is small, but also this term is small and this term is small. Right? But what happens to n? Huh? What is n? Is the population of bacteria. And so initially it's equal to is equal to a constant, right? With a little bit more excitement. It's equal to a constant, right? So if it is equal to a constant, what we have here? We have two equations where initially we can approximate, right? We have this virus that arrives is at such a small density that doesn't impact the growth of the bacteria. Thank you. The phages are at such a small density that the number of bacteria is still the ones that was before. So what we can do is that this one we can approximate it with n star. Okay. Right? And now, what do you see in this system? Right? You see that you have one equation which is phi v prime, you have v prime, I prime, I prime, v prime, v prime. Right? So this equation is, it only depends, these two equations only depends on v prime or I prime. So it's linear. It's a linear equation, right? We should be very excited now. Because it's a linear equation and linear equations are easy, right? Are easier.
Yeah.
Okay. So how this equation looks like? So the equation for, we can write d dt of I prime v prime as a matrix, okay, multiplying I prime and v prime. So what is this term? This term is going to be this term here, minus eta plus omega. Okay? Then we have this term here, which is phi n star. Okay? And then we have this term here, which is, you tell me, because you say yes. What is this term? Beta eta, beta prime eta. Okay? And what is this term here? It's the other two terms, right? It's minus omega minus phi n star. Okay? So now we have an equation like this. What can happen to this equation? I mean, none of you, I know that not all of you know, but some of you do. Okay, what's that 2?
Equal to 0.
Well, this equation equal to 0 means that these two are 0, right? Here you have a linear equation. Suppose that you have, this is like a linear equation with two variables. Suppose that you have a linear equation with one variable. Okay? You have d dt v equal to a v. What can happen to this term here? If it's positive, it grows, right? If it's negative, it doesn't. Okay? So if you have a one-dimensional linear equation, you have two outcomes. Either you explode or you go to 0. Right? This is what we started from. Okay? Now this is the generalization to this. We have two variables, right? But it's still linear. What can happen is that either goes to infinity or it goes to 0. What determines whether this blows to infinity or goes to 0? Yes. But what about the matrix? Which is not the movie. Huh? It's not the initial population. This actually is interesting. It's independent of the initial population.
So this guys.
Huh?
We actually talk about this.
Yes, I know. The eigenvalues. Okay. So if the eigenvalues of these are positive, if there is one positive eigenvalues, these will blow up. Right? What does it mean that these will blow up? It means that the population of the new viruses will increase. Okay? If the eigenvalues here are negative, the population of the new viruses is going to decrease. Okay? Do you agree? Yes. Good. So, what are the eigenvalues of this matrix? We hate doing this, but there is one trick that you can do when you have two-dimensional, when you have two-dimensional matrix, which is that you can look at the trace and you can look at the determinant, right? And the trace is the sum of the two eigenvalues. The determinant is their product. Okay, so we have two eigenvalues. We want that one of them. I mean, we can see that the trace is negative, right? So the sum of the two eigenvalues is negative. We want one of them to be positive. So negative. To have one positive, their product has to be negative. So we want the determinant to be negative. So we have this matrix here. There are two eigenvalues, lambda 1 and lambda 2. The trace of this matrix is this number plus this number, which is lambda 1 plus lambda 2, which is negative. Then we have the determinant. The determinant is lambda 1 times lambda 2. Okay? So this is negative, so at least one of the two has to be negative. If we want them to be, if we want one eigenvalue to be positive, we need that this product is positive. Is negative. Okay? So what is the determinant? Well, the determinant is eta plus omega times omega plus En star minus beta prime eta En star. Okay? And we want this number to be negative. Good. So then if we substitute, what we want is beta prime eta times Tn star over eta plus omega to be larger than omega plus Tn star. Okay? So if we substitute, we get beta prime eta this term here, it's omega over phi. And then we have 1 beta minus 1 minus omega. This has to be larger than this term here, which is omega plus beta plus omega times omega, beta minus 1, beta minus omega. And this. So this term here is equal to, I don't want you to follow the calculation, just to follow the flow. So it's minus beta minus 1, beta minus omega times omega plus beta plus omega times omega divided by these things. That simplifies with this one. And then here you see that this term becomes beta, beta, and then of course I forgot some p-value here. Yes, here was divided by, here there was not p because it multiplied. So here I get beta, beta times omega. And so if we simplify this equation, what do we get? We get on this side beta prime larger than beta. So what we discover with this calculation is that the population of the invader, now it was fun if I was turning around and there was nobody. That was pretty fun. But so what we discover with this thing, okay, is that basically we have an invader that grows if what happens? If beta prime is larger than beta, which is exactly what we started from our intuition. Okay? So what we see is that an invader will grow, will expand in the population if beta prime is larger than beta. While it declines, it has a negative growth rate initially, if it has a beta prime lower than beta. Okay? So what we can do is that we can make this systematic, right? Now, imagine in a simulation, so questions about this? Is this sufficiently unclear? Okay. So what we discover is that in this case, the growth rate of the invader, the rate at which this invader grows, is something which is proportional of beta prime minus beta. Okay? So the difference between beta prime and beta determines how fast this will grow. Okay? So what you can do to make this more systematic, which in this case it's pretty trivial, you can run a simulation where you, for each value of beta prime, and each value of beta, what you do is that you check what is the file. So for this point here, what you do is that you have your population, the invader, you run a simulation, and you check what is the value here compared to the initial value. Right? So you check whether this grew or declined. Right? And what you see is that this number here is big if beta prime is larger than beta. Right? And this small if beta prime is less than beta. Right? So this is in the code that you have if you want to run this. Okay? If you want to make it even more apparent, because you see that you have like some shaded line here, you can check whether this is larger than 0, or so this difference, sorry, is positive or negative. Right? Meaning, did it grow, or it declined? Right? If you do this, what you see is that, well, in this case you see the same, you see that it's yellow above, and it's purple below. Okay? So what does this mean? It means that, how do you interpret this plot? The way you interpret this plot is that you say, suppose that I have a resident population that is at some value of beta. Right? And then there is an invader that arrives, which has this beta prime. Okay? So you have this beta, and then you have this beta prime. Will this invader be successful? Yes. If I have this value of beta and I have an invader here, will it be successful? Okay? Is it clear what happens, what is the interpretation of this plot? And now you can see that this plot gives you an intuition of what is the evolutionary dynamics over a longer time scale. Why? Suppose that I have a population, I start with a population where there is an individual, I mean there is a population and it's here. Okay? And now there is a mutation. And there are a few individuals that have one individual that have a different beta prime. Okay? So it's here. What is going to happen to the system? What is gonna do this invader, this mutant? It will grow. It will grow and displace the old one. So what is gonna be the new resident population? Yeah, so I have to take this beta prime and copy it to a new beta. So you see that you move from here, this invades, so the new beta is gonna be here. Now suppose that there is a mutant that has a lower beta. What is gonna happen? Nothing, because this mutant will not expand in the population. And so you stay here. Now, suppose that there is another mutant here. What is going to happen? It will invade. Right? And so the new population is going to be here. Now, there is a new mutant. What is going to happen? Invade. So what do you expect from these over evolutionary times? So if you wait a long time and you look at your system, what do you expect? You expect beta to grow, right? Because there are new mutations. These new mutations, when beta increases, they invade. And so beta in the population will grow. Is this clear? Good. Okay, so let's make things a little more spicy. Okay. Good, so basically what we discovered here is that it looks like that, here I use optimal, but optimal is not, anyway, that over evolutionary times, evolution maximizes the burst size, right? That if we wait a long enough time, what we expect to see is that we are going to see higher and higher burst size, right? This is our expectation. Yes?
The assumption that the number of cells is constant.
Yes, this is an interesting point, which is a consequence of this. Actually, one interesting consequence of this is that what you expect is that over evolutionary time, there are going to be fewer bacterial cells. But what you're saying is we were sort of assuming that the number of bacterial cells was constant. It's true, but we are assuming that, so we are looking at two different time scales. Right? We say we are here, and there is a mutant, right? And then there is some time that it takes for the mutant to displace the other one, and the population of the bacteria will change. Right? And then we are here where there is a new population, right? Which is static. The number of bacteria is different from before, but it's static. And then we have a new mutation, and then we check what happens. Okay? Is this clear? What is unclear? Let's change the question. Okay. So
There is any what happened
Yes, one interesting consequence of this is that we saw that in our calculations that the population of bacteria was something like omega over phi eta plus omega over beta minus 1 eta minus omega. Okay. So what happens at a certain point is that you see that this term here, right? If it becomes bigger and bigger beta, the value of n star will become smaller and smaller. Okay? But this is like sort of expected in a sense. But another way to think about this is that in the system there are, from the perspective of phages, there are some resources which are bacterial cells. Okay? And not all the bacterial cells are infected. The phages which are better are the phages that are able to infect more bacterial cells. So you expect that the number of bacterial cells over evolutionary times will decrease because phages are better off in infecting the cells. So what you expect over evolutionary times, not over ecological times, over successions of invasions and extinctions, that the number of bacterial cells will decrease as beta will increase. Okay? Good. So here the situation is interesting, but it's pretty boring, I guess, right? What you say is, okay, basically, if you produce more cells, you grow it. I mean, when I asked you the first time, this is the answer you gave me, and this is what happens. Okay. On the other hand, if you look at different phages, what you see is that the birth size can vary a lot. Right? Can vary from 10 to the 2, 10 to the 4. Okay. Now here it's plotted as a function of the ratio between the genome of the host over the genome of the virus. But even for a given, so basically, how big is the host genome over how big is the viral genome? Right? But even for a given value of these, you see that there is a big variation. Right? This is 10 to the 2. This is 10 to the 3. Okay. So you see that it can vary upon an order of magnitude with some conserving this thing. Okay. So the question is why? Why don't we see stuff that has a burst size of 3 billions? Our model predicted that what is optimal is to increase the burst size. If you produce more viruses, you're better off. Why doesn't it go to infinity? Yes, but I mean, our model predicts that even with the finite number of cells, that you want to increase this burst size, right? And if there was a limit given to that, we would expect this number to be equal to that limit.
What's the x-axis again?
It doesn't matter here. This is like the ratio of the genome length of the host, of the bacteria. These are different species of phages infecting different bacteria over the length of the genome of the phage. So the idea is the bigger is the genome size of the host with respect to the viral phage, the more cells can produce, because basically, yeah. This is not my point. My point is, even for a given value of this, you have variation. So let's go back and can I start? Okay. And so this is, let's say that this is a okay. So let's go back to this thing here. Okay. And think about what's happening here, right? So imagine that you want to produce more viruses. Okay. What do you have to do to produce more viruses? Yeah, you infect, right? And then you have to assemble these viruses. Right? So suppose that you want to produce 10 viruses. Right? How long is going to take? Something. Suppose that you want to assemble 100 viruses. How long is going to take? A little bit more. Right? And so what that a little bit more is going to impact in our model? You can do it. It's not, yes, but indirectly. So there is one parameter which measures how much time is taking to go from infecting a cell to make the cell explode. What is that parameter? Eta. So what we expect is that if you want to have a bigger burst size, then it's going to take longer to build these cells, to build these bacteria, to build these viruses.
So at the end, beta and eta are like beta is a little bit like independent.
Yes. So the idea is that beta and eta are not two independent variables. If you want to build more, if you are a virus and you want to have an higher burst size, you need to spend more time inside the host, because to produce one virus is going to take some seconds. And if you want to produce 10 more viruses, it's going to take 10 times this some amount of seconds. Okay? And this is what you can observe. Now let's not focus on the reaction, but what you can observe is that the latency period, which is the time it takes here, which for us is 1 over eta, okay, roughly. So the time for us is 1 over eta, because eta is a rate, and this is beta. So what we see is that if you have a high beta, you have a high latency time. So you have a low 1 over eta. Okay? Good. And so these two things are linked. Okay? And now there is one other observation. One other observation that you can, no, I'll show you later. Okay, so let's look at this. So how do you include this effect? So the effect that beta and eta are not independent. What do we have to do? So now the new phage will have the same value of eta as the resident population. Huh? No. Right? What we'll have another value of eta. Right? So what we expect is that if we write the equation of the invader, this invader will have some E and E prime minus eta prime plus omega I and dv prime T is going to have minus eta prime I prime minus omega d prime minus T and right? Sorry, beta prime. Okay? Where now not only beta is different, but also eta prime is different. Okay? Is this clear? Right. And now we have to decide how do we choose beta prime and eta prime. But if we choose them independently, what do you expect? Suppose that you don't have this constraint, that you can do whatever you want. You can have the arbitrary high beta prime, arbitrary low beta. You can have whatever values you want. What do you think is going to happen? Beta prime, what is going to happen to beta prime? What do we see earlier? It will increase. What is going to happen to eta prime? It will also increase, right? Because, I mean, why don't you have this Darwinian monster of a virus that is produce a lot of viruses and it takes 1 picoseconds to produce them, right? The point is that there are constraints or trade-offs, okay? So you cannot, if you are a virus, you cannot produce 10 to the 5 viral particles in 1 second, okay? It's going to take a bit longer. So what we can imagine is that beta and eta are a function of each other. Okay? So for instance, beta is a function of eta. Now what function we want to choose? Well, we want that as function of eta. So if eta is very large, so you are very rapid. Now eta is the opposite of the latency period, right? So it's something that tells you how fast you go from infection to burst. Okay? So if this number is very large, what is going to happen to the batch size? Huh? No. Decrease. Because eta is the rate, right? So it's how fast you are, the speed. Okay? So if you are here, this will decrease. And then as you decrease eta, this will grow, right? And then we can say there is a maximum, because still, I mean, viruses occupy some amount of space, a cell is some amount of space. You cannot have infinite viruses inside the cell. There is some maximum beta naught, okay? So the idea is, if you are very, very slow in producing viruses, you can produce a given amount, a maximum. If you become faster, you have to produce less, right? So these things, if we want to write these things in one way, we can say that there is something of this form. Okay? So beta naught is the maximum, and eta naught is the latency rate at which beta is half of the maximum. Okay? So you have some expression like this. Is this clear? Good. Okay. So let's do this. I'll say one thing because I think that the tension is going down. So, and I'll continue tomorrow. So what do we do with this now? Huh? What does it mean, solve it? This is not an answer.
We substitute the function of
Yes, we can substitute the function and say that we have, well, we can invert the function and say that eta is equal to eta naught beta over beta naught over beta minus 1. Okay, and we have this equation here if you want. I mean, these two quantities are linked, that's okay. But then what do we do with it? Simulate it. Yes, but that's not the answer. Yes, we can simulate. You will simulate it. But how do we know? Suppose that we have, I mean, we learned earlier that we can simulate, but there is actually a very good way to understand what is going to happen, which is to look at what the invader does. So we assume that we have a stationary population. Okay. So we assume that we have some n star equal to the stuff we calculated before, which is going to be omega plus eta over beta minus 1. If you want beta of eta minus 1, eta minus omega. Okay? And this is the stationary population. There is one type of viruses with a given value of eta and beta, which are linked somewhere here. And then there is a stationary population of the bacteria. And then what do we do to understand what evolution does? What do we want to know about these new viruses that just arrived? Huh? Huh? What do we want to know? We want to know. Yes, we want to know. So suppose that we have, imagine it as an experiment. Right? Is what we did here. Imagine it as an experiment where you start with some population of viruses, some population of bacteria, and then you introduce a new mutant. What do you want to know about this mutant? If it grows, exactly, if it grows or declines, right? So how do we get to know about whether this new mutant grows or declines without simulating the equation? Yes, so I get values of what? Of which system? What do we have to do? There is one thing I have to do, and then you're free. What do I have to do? I mean, these things depend on n. The n dt equal to blah, blah, blah.
Express it again as a linear equation.
Yes, so what I have to do to this n?
And
And star. So I have to assume that this mutant is at a very small population, doesn't affect the how many bacteria there are, and then I have to check whether it grows or not. Okay. So how do I check now? This is a linear equation. How do I check whether this grows or not? Huh? No, not the fixed point. This is a linear equation. There are two options: it declines or it grows. Yes, with the eigenvalues. Right? I check whether these things grows or these things declines. Okay. Good. We're gonna check it tomorrow. Okay. Good. Now think what is gonna happen. Okay. Good. See you tomorrow. I mean, see you now.